All right. Maybe we can get started. I am not sure. I think we still will have people arriving, but in the meantime, we might as well get started. So welcome, everybody. My name is Sheila Heymans. I'm the Executive Director of the European Marine Board, and this is the European Marine Board's third Thursday Science Webinar series. We are recording this, and it is going to be live streamed, or it is actually being live streamed. So please act accordingly, I should say. If I go to the next slide here, just some housekeeping rules. Please make sure that your name is clearly entered so that when we ask a question, we know, when we read a question that you've asked, we know who you are. And please use the Q&A, which you should find at the bottom of your screen for your questions. And when you do, please also put in your organization and country. That's always helpful. And I will read the questions after the talk and ask the questions to the speaker. If you have any technical issues, please use the chat function, but try not to use the chat function for questions. And as I said, it is being recorded and it will be made available online on our website and on our YouTube channel after the fact. So today's talk is actually linked to our new document, Navigating the Future 6, which is our flagship document that highlights the ocean's place in the world. The document will be launched, just to make some promotion here, on the 23rd of October in Brussels at the Talk CEC. It's a venue in Brussels. So if you are interested in to hear more about it, please do register to your register to be able to attend there. So without further ado, just to say that this webinar is linked to the chapter on ocean and fresh water that's coming out in this document. And our speaker is João Frias, who is a senior researcher at the Marine and Freshwater Research Centre of the Atlantic Technological University of Ireland. And he is a co-author of that chapter on the ocean and freshwater. So, João, maybe you can put your camera on. Maybe your camera's on already. It is indeed. And I'm going to stop sharing, then you can start sharing and take it away.
Super. So just 1 second in here. Are you able to see the screen?
Yep. Yeah. You see the screen and it's in full screen mode, so it's all good.
Perfect. Great. So good afternoon, everyone. I would like to start by thanking the invitation of the European Marine Board to present a little bit of my work on plastic pollution, and more recently on the work of an international project on the plastisphere that enabled my contribution to Navigating the Future report. Today, I will talk a little bit about international travel. It is quite suitable because it's summer. Many of us are thinking or actually traveling, and kind of depending on your lifestyle, you've been in one of these situations where you either are hitchhiking a ride to a place where you want to go, or you already arrived and you're looking for your bag in a busy airport or in a bus station, which is the perfect analogy for what happens with marine litter in the ocean. Marine litter is comprised of a diversity of materials. However, plastic accounts between 70 to 90 or more than 90% of all the marine litter items found in beaches and in coastal areas. And once in the environment, plastics are colonized by a diversity of organisms, and also plastics can absorb persistent organic pollutants and trace metals, as we're going to see in this talk. So the hitchhikers are bringing unwanted luggage with their travels, with consequences that are not fully yet understood. Plastic is a very versatile material. It allows most of our daily activities, from research to transport, from clothing to art. But I like to make a distinction between essential and non-essential single-use items. So plastics are fundamental for medicine, for research and development, for activities that contribute to increasing our life expectancy. However, non-essential single-use items, cups, cutlery, takeaway containers, they contribute to a large waste management problem, which is particularly prevalent at sea. Most recent estimations point out about 19 million tons of plastic are leaked to the environment. About 1.7 million are transported from rivers to the ocean, which just highlights how big of a problem it is. And the scientific community is still trying to quantify what is happening. So today, I would like to take you on a journey with me, and we're going to explore some of the concepts and some of the specifics of a case study across the Atlantic Ocean and the Mediterranean Sea before diving in what we can do next. So let's start with how I got involved into this topic. And my journey starts in Portugal in the late 1980s, early 1990s, in a beach in Caparica, close to Lisbon, where I would go with my family. And as a child, I don't remember the beach being polluted with plastics. And in fact, this was kind of always in the back of my mind. So I went to old family photos to see whether there was evidence or documented evidence of plastic pollution. And I couldn't find anything on the photos. However, the reality was quite different when I went in the early 2000s to the university in the same area. And the beach that I used to go with my family was now covered in plastic, like we can see in the photo. So this brought my interest, and it started the early stages of my career, initially in Portugal with Professor Paulo Sobral, and then later in my PhD, also with Professor Paulo Sobral and with Richard Thompson as my co-supervisor. And I completely dove into this topic. I want to take you on this journey also on the concepts, because in my career, I went very fast from marine litter to microplastics. And instead of just providing you with the definition, I want to understand, I want to share with you the complexity of what is a microplastic. We have a lot of criteria for microplastics, and as you can see here in a paper for some colleagues in Denmark, microplastics are marked in aqua blue. Not everyone across the scientific community has the same definition, and this is just for size. So the first time that microplastics as a concept was used was in 2004, and it described microscopic pieces of plastic. And then this concept was further expanded in 2009, where upper limit was included in the definition, and this upper limit was 5 millimeters. The definition has been further expanded in 2011 and in 2019 to include origin, shape, color, and even other matrices. So for this presentation, when I refer to microplastics, I will be referring to any synthetic solid particles or polymeric matrices like films, which have regular or irregular shape, a size that ranges from 1 micron to 5 millimeters, which either have primary or secondary origin, depending on whether they were produced to be or to have microscopic dimensions to begin with, or if they result from the fragmentation of larger pieces of plastic, all of them that are insoluble in water. The definition has a lot of criteria, as again, the team in Denmark pointed clearly in their paper, and we can have a full list of criteria that could include or exclude microplastics under different frameworks. This is particularly relevant, especially outside of academia, as there are a diverse range of stakeholders who are investing resources to effectively manage plastics and microplastics. But as you can see here, even standardization bodies have different definitions on the concept, which adds to the complexity. And then when we look into policy, particularly to the United Nations legally binding treaty that is under negotiation, we can see that the definition for microplastics also includes nano-sized particles. So we really need to reach a consensus on the definition so that we can progress not only on research, but also on policy to the next level. And hopefully with the Plastics Treaty, we will have some clarity from next year onwards. The second concept is relatively easy. It's plastisphere, and plastisphere describes the microbial community that is attached to the plastic. And this community is different from the communities in the surrounding environment. It's a concept that is focused on the ecological structure of the biofilm layer, growing on the plastics. And it is supported by research into the formation of several ecocorona layers that are colonized by diverse organisms. Some of them might be potentially pathogenic organisms before establishing a substrate where other species and pollutants might be able to attach. Species such as the barnacles that we saw before. The plastisphere and its organisms can increase the toxicity of plastics and microplastics, as they have the potential to absorb persistent organic pollutants and trace or heavy metals. And in fact, we know they accumulate these pollutants. So in the early stages of my career, in the beaches where I was going in Portugal, I was really interested in the pellets, which are these round granules that you can see in this photo. For those of you who might not be familiarized with it, the pellets are the raw material to produce virtually any plastic that you have around you right now. And in the first stages, what I did in my master's was kind of looking to different beaches where we collected the pellets, and then we assessed for pollutants. And we were particularly interested in PAHs, in PCBs, and in DDTs. And from the data that we collected, we noticed that we could find these pollutants in the plastic. In the beginning, we only did it in 2 beaches, and then we came across this project called International Pellet Watch, led by Professor Hideshiga Takada in Japan, and we kind of like started talking with the team over there, and we expanded to a larger assessment of the 10 beaches across the continental Portugal, where we did the similar exercise. But this only is interesting when we compare with what is happening in terms of environmental concentrations in different matrices, such as sediment or organisms. And what we were able to identify was that the pellets had higher concentrations than the surrounding environment. So one of the facts from this research field is we know that persistent organic pollutants accumulate in environmental pellets at higher concentrations than the surrounding environment. So we started asking ourselves, so if pollutants accumulate and if plastics form biofilms, is there a relationship? Are the species affecting degradation or fragmentation? Does it interfere with polymer identification? Does it have any other biological implications? What kind of species live on the plastics? And we started like talking. This was like ongoing debate in conferences, and eventually we got funding to do a case study, and that's exactly what we did. This is how the microplastics project started. This project was carried out throughout the pandemic, and I can guarantee you that despite the many challenges that we faced, we were able to get enough samples and experiments across the Atlantic and in the Mediterranean Sea that support this presentation today. The project aimed to overcome some knowledge gaps in weathering and biofouling. We wanted to know how these processes influence fragmentation, distribution, and dispersal of microplastics in the marine environment. It was a very interesting project because we were exploring biology, chemistry, physics of the microplastics so that we could gather as much data as possible to improve models. And today I'm going to talk about a very specific case study that we did with in situ experiments across 5 countries. So in the Atlantic, it was Ireland, Spain, and Brazil. And in the Mediterranean, it was France, where we had 2 sites, and Italy. And basically what we did was we had different polymers, including one bioplastic, that were cut by hand. As you can see, our colleagues in France cutting the plastics for their own experiment. Each lab had to cut their own plastics. And they were introduced in this containers that were perforated so that the water could flow. And they were introduced into a cage which was deployed at sea for 1 year. And we were sampling every 7, 14, 30, 60, and 90 days. And once we reached the 90 days, we would just replace the plastics in the original container because we had been taking them for the different analysis that we were doing. This is an example of the deployment in Galway. We did the deployment in the port of Galway, and the cage was added to one of the floating pontoons to ensure that with the tide movement, the cage were always underwater, and we could do this to ensure that the plastics were always in contact with the water. In this case, it was done in the end of the river, kind of like between the river and the bay, so it could mimic a estuary area, but in other parts it was done under marine conditions. And this is some of the things that we've assessed. So starting above with the scanning electron microscopy from our colleagues in Italy, what we can see is the development of the biofilm layer and this eco-corona layer on the surface of the plastic. So we were trying to assess what happened in terms of like a biofilm growth. A team in France was assessing the biological diversity using DNA metabarcoding. The team in Germany was looking into spectral changes in FTIR and in Raman over time. And all the teams were identifying like the species that were attached to the incubated plastics, as you can see here on this example from species that we collected here in Galway, so that we could get as much data as possible. And what have we found? We found that trace metals accumulate in the biofilm from the surrounding environment. So what we were analyzing was 12 different trace metals in the biofilm, and we've had one biopolymer because we wanted to see if there was some sort of difference between the biopolymers and the traditional fossil fuel-based polymers. And from our study, it seems that they behave similarly. In fairness, PLA in some of the cases had higher concentrations than the fossil fuel-based polymers. I can tell you that both the cleanest site, which was Villefranche in France, and the most contaminated site in Naples in Italy were in the Mediterranean. And one very particular interesting thing of this work was that it was the diversity of the plastisphere and not its biomass that seemed to influence metal bioaccumulation. As I said, we were exploring all different kinds of things. So we also assessed the holes that exist on the plastics because this is fundamental to improve dispersion models and prediction algorithms. This happens because the holes can accumulate organisms, can accumulate sand, can suffer abrasion. Eventually, there might be like a connection to fragmentation, and this is valuable information when we want to understand the dispersion of plastics, particularly to deeper marine layers through either natural deposition or through marine snow. As I was saying, we also assessed the biological diversity. Over 550 biofilm samples were processed for viruses, bacteria, fungi, and other planktonic organisms. However, because the manuscript is still in preparation, I cannot share more details at the moment, other that a wide variety of organisms were identified, particularly between the North and South Atlantic, and then between the Atlantic and the Mediterranean. And it seems that Toulon and Galway had more prokaryotes and Naples had more eukaryotes than any other site, but we will have more details on this pretty soon. However, I can share one interesting hitchhiker that we found on the plastics, which were tardigrades. Some of you might know them as water bears. And this was the first time that we found this specific extremophile organism that can adapt to virtually any environmental condition. And out of all the places that we had, and please remember, we had places in the Mediterranean and even Brazil, we only found them in the summer in Galway. Maybe came for a festival, I don't know. This is the first time that we reported tardigrades in the plastisphere, and this brings more questions than answers, actually, because in itself is an interesting discovery, but it opens the door to assess the prevalence of such species, and because we know that plastic is lightweight and it floats, even the potential to transfer potentially invasive species between continents. So it kind of opened another line of thought. And then on the work that our German colleagues were doing, assessing the differences, the spectral differences using FTIR and Raman, we kind of like could understand that after 60 days underwater, the FTIR spectrometry does not recognize the polymer when compared to the pristine database. So this highlights the need to constantly being updating the databases so that we can have clear identification. It highlights that some of the environmental samples might have lower percentage matches in the databases, particularly because of this, and maybe the need to apply forensic science techniques to how we are processing the data, which again goes back into the discussion of having standardized methods, but it's quite a challenge within the scientific community at the moment. On this note and on this specific data set, I want to share that our German colleagues, and you can see Robin Lenz in the photo, he created the data set and made this great app that is downloadable and accessible in Zenodo. And he was awarded the Fair for Chem Award 2024 in the NFDI for Chem conference, which just shows and highlights the importance of having such a tool to assess weathering of plastics and to improve the data for models. I know that I put like the links for it, but if you want to have access to all the papers and data sets, you can use the project website, and you can go and you can download everything. So on the next steps, of course, for the project, we want to publish papers. For the next steps within this field, we are looking to seeking partnerships, additional funding for ecotoxicology studies, but also to raise awareness within the scientific community as well with different stakeholders, so that we can have better understanding of what is happening. And beyond the scientific community, the next steps is to have talks and events like this to disseminate results in a way that it's easily understandable to audiences, but also to promote change, either in terms of behavior or in terms of policy. And in the beginning of my talk, I was telling you that plastic is a very versatile material, that if it wouldn't be for it, our daily lives and processes would be more challenging. And I wanted to point out that we need to understand that there is no way in the action of throwing away. In fact, going back to the numbers that I was sharing, we know that about 0.5% of global plastic ends up in the ocean, but 23% of the plastic waste is mismanaged and littered, and about 5% is leaked into the into the environment. So we have here some sort of accounting problem that it's quite evident when we look at the recycling data around the world, and we know that less than 10% is recycled, and this should raise a concern in itself, particularly when we know that plastics can accumulate persistent organic pollutants, trace metals, and more recently, we know that they can accumulate potentially pathogenic microorganisms. So if you're interested in this, I highly recommend you reading the Navigating the Future report, where we explore this and many other topics within climate and freshwater and biodiversity, and even the relationships between ocean and humans. I want to take this opportunity to acknowledge my funding institutions, because without their financial and logistic support, our research would have not been possible, particularly JPI Oceans, Marine Institute, and the Department of Housing, Planning and Local Government here in Ireland. And to thank the wider team and the wider consortium, particularly because they were really fun to work with during the pandemic. I want to have like a special thanks to Captain Brian Sheridan for allowing us to use the Port of Galway as a site during the pandemic, my colleague Colin for diving for this project, and of course Professor Wang Hongxi in Shanghai for this collaboration in terms of like exploring the different densities. And just before passing the word and opening for the Q&A, I want to leave you with this thought. When we think about the ocean, we tend to think about the partition that it's closer to us, either the Atlantic, the Indian Ocean, the Pacific, the Arctic. But we need to start thinking of it from a different point of view. We need to see it as one common resource. And we should manage it collaboratively instead of competitively. Because, you know, just because you don't see it doesn't mean that plastic isn't there. We know now, because we have robust data sets for decades now, we just don't know yet the effects in terms of like environmental health or human health impacts. And you will see a lot of talks of people within the plastic pollution field talking about or advocating for change. And we don't want to be like Cassandra from the Greek mythology. You know, she was a person whose valid warnings and concerns were disbelieved by others. And you know, if you like movies like I do, and if you like sci-fi movies, you know that every disaster movie begins with a scientist being ignored. So if you know a scientist and you don't understand what they do, have a conversation with them, understand why they are passionate about the research that they are doing, because you might find out that they're trying to make the world a better place for everyone, including themselves. So, Kore Mila Mahagat, thank you for your time today.
Fabulous. Thank you very much, João. That was a very nice way to end the talk. So we have a question in the Q&A, I see. And the question is, are you talking about true microplastics, or are you actually talking about nanoplastics with regards to size? Did you put size classifications in the
Yeah, it was because there is there is some sort of confusion even like with definitions. So when we're talking about microplastics, I'm talking about everything below 5 millimeters in diameter. And according to the current definitions that we have, some definitions say that it goes up to 1 millimeter. The majority of the definitions go to 5 millimeters, but we're not going into the nano size, because nanoplastics behave completely different than microplastics. So, and it's completely out of my field of expertise as well. So I wouldn't be the most indicated person to share a little bit of this information with you guys.
And then I see another question that came in that was quite similar from Rajari. Sorry, I'm really not doing the name any justice, Sahu. And it is, what is the convenient methodology to identify and classify the microplastics? And I guess that's what you were just saying. There are specific methods. Yeah.
So to identify and classify, we're kind of moving away from the solely visual observation because there's a lot of bias there, and we're using like other techniques like FTIR, like Raman, if we want to assess the polymer type. Also, you can use pyrolysis GCMS if you want to have mass associated with it, because and you will see a lot of this on the papers from the scientific community. We highly encourage people either to report the concentrations of microplastics per area or per volume, or if you don't have the number, to have the mass, or to have microplastics in kilos of dry sediment. It depends on the matrix that we are assessing and exploring. Yeah.
Okay, and then I see Peter Dowling has not followed the instructions and put the question in the chat, but I will give you this one, Peter. Two questions first. What plastics were surveyed for accumulation of biofilm, and is there a change with the use of different plastics?
So
yeah, maybe we'll start with that one.
Yeah. So the plastics that we used, it was a wide range of polymers. Basically, the most used low density and high density polyethylene, PET, polystyrene, and then we had on polypropylene and on PET, we had pristine and we had recycled. They were all made out of the same batch in the same company, and they were shipped to all the different partners. So everyone had the exact same product produced under the exact same conditions. And we had, of course, the ones that are widely used according to the report. So polyethylene, polypropylene, polystyrene, PET. PET is what it's made, the bottles, the water bottles, and polypropylene is usually the lid. So we had the majority of them, but we wanted to have a biopolymer to see if there was a difference, because they are currently in the market labeled as an alternative to traditional fossil fuels. PLA is one. There are dozens of different biopolymers, and I think that we need to expand this kind of like research field to include more biopolymers to kind of assess what happens in the environment, because under lab conditions it's one thing, under environment it's a different thing.
And then the second question from him is, what is the rate of deposition of heavy metals biofilms when compared to other common marine materials? So compared to, say, metals or wood or concrete?
That's a good question, and I don't know the answer to that, but it would be good to look into it. I think it would be a challenge for metal. Maybe this is better to ask my colleagues in Toulon, because they're more familiarized with this. But I think that even though metal is part of marine litter, it would be a challenge to assess trace metals on metal, because you would have a bias on contamination. I'm not sure if the method is possible to do like that. Yeah.
And then in the Q&A, because I'm going to go back to the Q&A, that was what we asked you to do. There's a question from Chantal Rockerman to say, you're looking for collaborators, and have you thought about working with Sea Shepherd or Greenpeace or other NGOs who have ships and could help you with the sampling?
No, we haven't looked into this specific potential collaboration. I think it's good sometimes to leave the realm of academia, because sometimes we're kind of locked in an ivory tower. So it might be a good way of doing it. Yeah, we can look into it in the future. Yeah.
And then Mr. Miss Sahu, who I'm not going to try the name again, asks, is there a change in the microplastic after antimicrobial resistance in microbes?
Is there a change?
Is it enhancing antimicrobial resistance in microbes?
So we don't know, because we're assessing it. We're really at the early stages of this type of research. Because I think that the first time that even the concept of the plastisphere was used was in 2013. So I think the majority of the people are using like DNA metabarcoding to assess what it's there. So we're kind of still just having a sense of what it's there. That's a very fascinating question. I think that's a question that might come follow up in a different research project, because we also find organisms that are not pathogenic. We found all types of organisms. The fascinating thing is that they concentrate and they accumulate because it's a nice substrate for them to be. The issue that could be potentially concerning, and I want to emphasize the could, is that plastics might act as surfboards across continents, bringing species that weren't there originally, and we don't know the consequences of it. But there is so much change already. And if we think like of, for instance, ships that travel across the world, that they might be arbored in different arbors along the way to transport whatever it needs for our daily lives to use. What happens to the biofilm that grows even on the ships? Like it's a very fascinating thing. I think it would be different because it's not on plastic, and we don't know. But yeah, you're giving me more ideas with this Q&A.
Well, that's the nice thing about it, isn't it? And then Gary Kate asks, what in your opinion is a primary concern with microplastic microbes? Is it the associated pathogens, invasive species vectors, or the weathering and affected transport changes?
So I don't want to really go into the results of the paper that Ana Lacerda is writing at the moment. I think that in some cases, some of the species that we found, and some of the bacteria and viruses that we found, they might be associated with specific circumstances that are happening even around the globe. There is a concern on, can these species stay there, and what kind of impacts would they have in terms of like environmental or organism health when they are at sea. This is particularly relevant, let's say, for the case of Europe and Northern Africa, because the Mediterranean is an enclosed bay. I think, like, because some of the organisms, let's use the example of the barnacles that they attach, okay? We don't know if they are causing like fragmentation and releasing more microplastics of a smaller scale to the environment that might affect other organisms. I know that I'm talking about a lot of unknowns, but maybe we need to look into these unknowns so that we can have a good mass balance exercise of what is happening in the environment.
Yeah, and I mean, if I may add my 2 cents worth there, I would imagine that actually all of these things are probably important to some degree. And in some cases, invasive, their vector as an invasive species might be more important than in other cases. So I think actually it's a difficult question to answer.
Yeah. And then you have to see, like, when you fragment, because I was talking about like trace metals and persistent organic pollutants that attach. But what about if the substrate itself has additives that might be released from this scratching? We don't know at the moment, and a lot of research is being put into these by teams all around the world. And certainly there are people who know way more about this than me. But yeah, just
I also understood that there were, in plastics, there might be some chemical esters, some chemicals that actually could have harm to human health, and that could be another thing to think about. That maybe, I'm sure there are people working on it, but
yep.
yeah, another thing.
I do have to say, if you have questions like this, the scientific community is meeting in September in Lanzarote for the Micro 2024 conference, and pretty sure there will be experts there that can answer this. This is
Yeah,
a free conference, so
yeah.
I highly encourage you
Yeah.
to attend. Yeah.
Okay, then we have, I think, 3 more questions. Another one from Chantal Rockerman saying, do you have recommendations for the plastic treaty being negotiated at the moment? Are you guys at the project level proposing?
I know that there is a coalition of scientists for an effective plastics treaty, and they are putting some recommendations out there, kind of to look to the whole cycle, like to really make it like a circular economy model, kind of like to really assess the additives. I don't know, there is a lot of things. I'm not directly involved in the negotiations because I'm a scientist. I'm not part of everything, but I know that within the scientific community there are people that are involved and participating and sharing the information and like trying to minimize miscommunication,
Yeah,
misinformation, and this kind of things. Yeah.
yeah, yeah. Okay, then there's a question from Joanna Rodriguez saying the work that you talked about is the first one demonstrating evidence of invasive species introduced to other places as a consequence of microplastic dispersion. She's asking
I,
if it is. I doubt it is.
I doubt it is as well. I think that that have been like documented evidence of like even I think in the 60s or in the 70s, I remember like seeing papers that had like uh, floating wood that would already have barnacles. It's not a new topic. Maybe it's a new substrate with plastic, but maybe this thing have been going on for decades and it's just been unnoticed. I wouldn't say it is the first time for sure. Yeah.
Yeah, but there is probably, maybe in the macro scale, it's, I would imagine, whether a barnacle or this barnacle attaches to a piece of wood or a piece of plastic probably doesn't matter. But biofilm, I suspect, would be it would be easier for it to accumulate on something as sort of smooth as plastic, whereas wood tend to be a bit more rough. So I mean, there could be differences. But indeed, as I understand it, this is not the whole invasive species being floating around on other things, whether it's just a metal container that's floating around or whether it's plastic. I don't think that makes a difference. It's just so much more plastic now than other things floating around in the water before. I think that's the point, is that it's there's so much more rubbish and plastic is so much more
prevalent.
Yeah, buoyant. Yeah. Sorry, you were saying.
No, I was going to say I agree with you. Maybe there might be a difference between the organisms that attach to one matrix or to the other, because of the conditions of the material itself. If you have a metal, maybe not all species would attach to it. If you have wood, maybe it's more closer to what the organisms are used to. So I don't know. I really don't know.
Okay, then an anonymous attendee is asking, the outlook on plastic pollution is not very promising. If this continues, the problem will become increasingly troublesome. Besides generating data and publishing articles, should scientists play a more active role in this matter? So should we advocate? What do you think about the upcoming plastic treaty that is expected to be announced by the UN?
So, two questions. Maybe the first one is if scientists should play a more active role. Scientists are playing quite an active role with the Coalition for an Effective Plastics Treaty. There's quite a big group of people that are involved in the processes, involved in the dialogues, people with a lot of knowledge, a lot of experience, a lot of expertise that can contribute to add a very positive or a more positive outcome from what it might be the end result of the document. So on that, the data that has been generated and the papers that have been published are actually quite a good add-on and a contribution. That's why also this report, the Navigating the Future, is also important for us to be putting the information, not only like the scary numbers, let's put it like that, the statistics and everything, but kind of to open the mindsets for the people who are the policy makers or the decision makers. I think it needs to be a constant dialogue so that information doesn't stay like in an ivory tower, and that's like the first part of the question. On the upcoming of the plastics treaty, I think that it's kind of like if you would ask a question on nearly anything, if someone likes something, you know, you will have 1/3 of people that are very positive, 1/3 that are very negative, and 1/3 that are neutral. I think that the key is the neutral. If the neutral are informed, they might be able to make it a more positive outcome. So it's really about the quality of information, the quality of data, and really looking at it not with the same mindset that created the problem, but with a mindset that can actually solve it. And I do have to say, there are a lot of collaborations with industry. Industry has opened also the support, the minds. There is good collaboration. There is collaboration that is a little bit more challenging, but you know, that's our history as humans. You know, there are periods that are challenging, there are periods that are quite smooth. It's just making sure that the document, the final document, will have a positive outcome for the people that haven't been born yet. You know, really going back to the definition of sustainable development from the Brundtland report in the 1970s, and really focusing on meeting our needs, but also allowing future generations to meet their needs when they need it. And I think for that, we all need to reduce a little bit our lifestyles, particularly in the Northern Hemisphere. But anyway, that's a whole different topic.
Yeah, if I may just add on to that, I think the first question about should scientists be doing more? I think I completely agree with you. Scientists are doing a lot already. We might not all be out there speaking to the television or, you know, making the case, but not everybody has the ability to do that well. And if you have a lot of people trying to do that badly, you're not going to help your own case. So I think the scientists must do the good science that's needed to support the cause, in this case, you know, reducing plastic pollution, making sure that people understand the impact of it, people understand the full implications and on all of it. And then you need good communicators, or I mean, like the Marine Board is doing here. We're basically taking all of this information together, put it together, and try and make it understandable to policymakers and the general public. So that is a function that we do. That's not to say what the scientists do is not important or that they're not doing enough. They are doing precisely the right thing, but you can't expect them to also do the communication, because you know, people have expertise and people they only have so much time, so they should be spending their time doing the things that they're good at. So I think you're quite right. We need to all be working hard in our perspective fields to make sure that this is something that's addressed. There's no more questions in Q&A, but I do, Britt, because we can't actually put it in the Q&A, she also has a question more about if you have any ideas about the latest science on the impacts of microplastics on human health, specifically. Yeah, I mean, you know, through, say, uptake in fish, through fish, eating fish, or other ways. I don't know if you have more information on that.
So I want to start by saying that I'm not an expert in this field. There are occasional papers that explore and you probably have seen it because they always make the media, that we found microplastics in the lungs, that we found microplastics in the blood. There are some more concerning, which is finding microplastics in the placenta or finding microplastics in the testes. Those are slightly more concerning, but we're at the point where it's kind of like a tick box exercise. Are they there? Are they not? We're not assessing the effects of the consequence. Yeah. So it's not really yet at the level where we can understand. For human consumption, like on fish, I would say it's relatively safe because we don't eat the guts, which is where they would accumulate. Even like the livers or anything, we remove the guts of the fish. There are a lot of work being done in terms of assessing ecotoxicology. For me this year, it's kind of like a sensitive topic because one of the persons that was doing the majority of this work, which was Professor Dick Fatak, passed away this year, and he was kind of like the inspiration, the promoter that would be looking more into the environmental ecotoxicology side of things. At this moment in time, we don't know. I think it goes back to the standardization question, because we know that plastics have additives, but most of the formulations are patented, so they're not part of any database. And if they're not part of any database, how can we assess? It's kind of like having a fingerprint in a specific place and trying to look for a person, but all the databases are locked. So we don't have access to it. So it's a little bit of a inglorious situation. Inglorious is not the correct word, but you get what I mean. Of trying to figure out how to assess. Then, most of these things are dependent like on differences in salinity, in temperature. Climate change is happening. So it's just like we have to fine tune the variables, but we also need to understand most of these exercises are done in laboratory conditions, and laboratory conditions do not have everything that is happening in the environment, and doing it in the environment is extremely costly. At this moment in time, like there is like a little bit of information, and we're still trying to figure out from an ecotoxicology point of view what is happening. I don't know, and probably no one in the scientific community would be able to say this is a fact at this moment in time. But it is scary that we find it in places in the human body where we shouldn't find them.
One thing I will, moving a little bit away from that, but also sort of linking a little bit with ecotoxicology, I know that the JRC, so the European Commission's Joint Research Centre, is doing some work where they're actually modeling microplastics through the food web. So, they are looking at how it might accumulate in top species that you might be fishing. But indeed, that next step of what does it mean when you eat them? When you were talking about, we don't eat the liver, we don't eat the stomachs, I was actually thinking about fish meal. Because fish meal is basically created by all of that stuff, right? And that is then fed to aquaculture products. So, or chicken used to be, I'm not sure if they do that anymore, but they used to. So what is the implication of that, I wonder? And I don't know if anybody's looked into the plastic implications in fish meal, for instance.
No, I don't. I at least that I might be aware. No.
No, I don't. Yeah. So anyway, I don't see any more questions, but that was a great talk. Thank you very much, João. That was very, very inspiring. Maybe you want to put in the chat to everybody the website of your project again, because I missed it. I was going to stick it in Twitter.
Okay. Yeah.
Fast for me. And while you do that, I will share my screen.
Let me just.
I want to share.
I'll put also this.
What have I done now? Can you guys see? No, you can now just see my.
We can see the background, not
You
the
can see the
present.
background, yes. And I will.
Yes.
Right there we go. So thank you again. I will just end with saying thank you everybody for coming, and to tell you that our next webinar will be on the 19th of September. So we will take a little break in August on the skills intelligence in the offshore renewable energy sector by Lefteris Sarkopoulos. I'm sure I'm not doing his last name any good either, but I look forward to that. And thank you again, João. And for those of you who want to know more about the document, you can go online to our website to actually see when the document is released, and it will be both online and in person. So you are more than welcome to follow that online on the 23rd of October. So with that, I want to thank everybody again. Thank you again, Joe, and see you guys in September.